Methane. Pass through really easy, but what is methane? Hydrophobic, so it's non-polar, and it is small. That's a key. Size, shape, polarity. That's what dictates things moving through. Size, shape, polarity, okay? Something that's really large, not going to be able to make it through. Really weird shaped, probably not going to be able to make it through. Polarity, really strong polar, definitely not making it through, okay? So if I have an ion, is it going to make it through? No. Is things like glucose going to make it through?
No.
I have a question for you. On an evolutionary level, what would happen if your ions and glucose could pass through a semi-permeable membrane? Talk to each other. What could happen? It's a good exam question.
If iron and glucose could... What?
If ions and glucose could just enter the cell whenever they wanted, what would it affect?
pH, that would be affecting the...
Ions would affect the organelles inside.
The structures?
They would affect the structure and
The shape, of course.
function of the organelles and enzymes on the inside of the cell. How would glucose affect it, though? I mean, I think that's the same as increasing the sugar.
Oh, yeah.
All right, what would happen on an evolutionary level if ions could pass through? You'd be dead. You would never have evolved. What is a membrane?
It's a permeable...
It separates things, the environments, right?
protector.
It protects things from leaving. Do we want your glucose to leave?
No.
No. If your glucose is leaving your body, what disease is that? Diabetes, excellent. That's what that disease is. So, does anybody know how they used to test for diabetes? It's a very simple test. They taste the pee. Excellent. If you are diabetic, what will your pee taste like? Sweet. That's how they used to do it. Doctors used to take the finger, dip it in, taste it. The good news is pee is, for the most part, sterile. Still gross, but for the most part sterile. So it's not going to kill you, therefore it's a reasonable diagnostic technique. Yeah, we just have a test, okay? You just pee in a cup, which you're going to do next quarter, and test it. I won't have you drink it. It's fine. All right.
It's about to quit.
Adenine triphosphate is what molecule? Adenine triphosphate is which molecule? ATP. Can ATP pass through a membrane? No. It is gigantic. Phosphates are huge molecules, okay? They're huge, they're charged, it's polar. Is it going to make it through a membrane?
No.
No. So we're going to need specific channels to allow those things to pass through. So if you cannot pass through the membrane, you need a channel to be able to pass through. Okay? Keep going.
That makes sense. Yeah, I think it should be very fast. Who said Violet before? Violet. It was a test on Friday. Oh, I didn't see it. I would get nervous. Yeah, that's not my
No, I was close. I'm just lost. All over.
Well, we can work together as a team, you know. Just saying.
Yeah, it's just okay. So I see here the same ATPs,
Okay.
and then here, do these convert?
They
to-
converted. Excellent.
Okay, so then I have 3 here and 0 and 0. That wait, no.
Yeah, ATP used.
0,
Excellent. Yep.
0, 3.
Correct.
Oh, okay.
So what from the reading to, if you're using ATP, what are we doing with it?
I don't know. What are we doing?
What are types of transport?
Active transport.
Simple transport, active transport, facilitated diffusion, right? So if we're using ATP, this is active transport.
Active transfer. And how come this is outside 1.5? I didn't understand because I counted 6 outside of all of them, or in each one it's 1, 2 stars.
No. 1, 2,
Is it just relative?
3, 4,
to the
5,
other
6. Yeah, it's relative to each other.
Oh, it's just relative.
Have to call Mandy and ask exactly what it means, but you can ask her next year.
And what are the circles? Do you know?
What are the symbols? They're
Circles. Just
They're just
Whatever.
They're all just random. Like this is
Call them whatever.
lipid. This is an ion. Just they're all whatever.
but we don't know exactly.
Yeah. They're all just proteins that are getting transported or diffused.
What about these things outside?
So those are the channels.
That's what I thought.
So like they're like the protein enzyme channels.
So then you would say that it's transported into cell against its concentration. That would be this one, right?
So which so
5.
which one has a higher concentration in the cell than outside? That's how I understood it.
Oh, I thought that it's transported into the cell against its concentration gradient.
Yeah, so like what's moving to a so things naturally move to lower concentrations. So if you have a container separated by a membrane, the concentration is going to want to go to the lower concentration to make it more random and uniform. So what's moving, it's asking which one's moving from a low concentration to a high concentration.
Oh,
So if you look at the outside the cell ones, what's higher than any of these? So 5, these are all 5, 3, these are all equal or lower, 1.5. This one's higher than the outside concentration, so that means it's moving against the concentration gradient.
this one here.
Yeah, so cell C, that's
All right.
stars, are moving against the
So
concentration.
I have my membrane.
Oh,
Yeah.
okay.
Okay. PO4 phosphate. In biology, we call that PI. Inorganic phosphate. Phosphate inorganic. ATP, glucose, things like that. Do they want or can they traverse the membrane? No. So we need what across the membrane? A protein, a channel. Okay, and that channel is going to be specific. So if it's going to take phosphate through, phosphate has negatively charged, it's going to have a lot of positively charges on the inside to allow it to move through. If we move with the concentration gradient, so that means from high to low concentration, that's kind of what kind of transport? Simple transport, passive diffusion, high to low, or facilitated diffusion because we have a protein channel. High concentration to low concentration. If, let's imagine, this has a ton of phosphate already in it, but I'm greedy and want more anyways. What do I have to do? Use energy. Excellent. And use energy through what? Active transport. And how we do that is we take ATP and get ADP plus PI. Okay, that's active transport. So you have simple diffusion, passive transport, facilitated diffusion is passive transport kind of, and active transport. Active transport, you're going to use energy. Typically, We're going to put a big giant asterisk and say typically we use ATP for active transport. Okay? How I feel. Cells, certain types of cells really like certain things. For instance, your nerve cells really like having ions inside them. Okay? Your muscle cells really like ions inside them. So we're going to accumulate those through active transport. Okay, keep going.
So the steroids are for the muscles. Huh?
What?
Steroids are the muscle wants steroids. No.
Steroids are just hormones. That's all they are.
Oh.
Active transport definitely does, but I was wondering about.
The ions are like the electrolytes.
Mm-hmm.
So I guess if you're drinking like sports drinks that have high concentrations of electrolytes,
Oh, yeah.
you're increasing the outside concentration gradient, which makes it the active transport easier, I'm guessing.
I would think so, because that's what you're. In the case of the muscles, right?
Yeah.
Yeah, active is the one we're using for high. Because it's high. Yeah. The lower
ATP is synthesized in the
we just don't.
But we're not going to worry about that for like
Is basic mostly negatively charged?
Is what?
Is basic mostly negatively charged?
Basic?
Like high OH concentration?
I want to say yes, but I don't know.
It's for question 3. Because if the channel is negatively charged, they'll have a harder time passing through. But if it's positive, they'll be easier. So that's what I'm confused on which way to answer.
Well, you gotta look up what those molecules, what those amino acids are. That's what it wants you to do.
I guess I don't even get the difference. I feel like they're interchangeable.
So I stuck it where they were off that. No, they're the blues, so I know where she is and where these are. Don't understand.
There's like so many ways with words to
No, just because you lose it. You're so bad at moving stuff, putting stuff in the right spot. So
I'm not understanding you. But I'll let you finish.
What were you asking?
I'll let you finish and then I'll ask you questions.
Is it about this table?
It's about both. I was trying to do the other one. But don't worry about it.
it's C because we're using this change so that you can move across the street. Now you got to figure out what's
just so that it How would you get fully
You got it? Okay. Well, let's do that last one since you're already got it. I didn't understand. Like she said, look up the amino acids, right? Or the.
So that was for the last question.
Okay. But did it mention lysine somewhere?
Which
Oh, here. Oh, because it mentioned. I was reading all over here. Oh, okay. You discover a mutant protein that has a lysine. Okay.
So
Number one.
Number one. Why are there only hydrophobic amino acids in membrane spinning protein, single strand membrane protein?
No,
I didn't know the answer to
hydrophobic
that
is
one.
hydrophobic, therefore it's hiding from water, so you have to
Hydrophobic.
It allows the
Let's read the question.
I think they're asking the same question there. Let me.
It's in the membrane because it's hydrophobic.
or a red inside the cell.
You're hydrophobic, where are you at? Inside the
Now,
inside the cell versus outside. It's just looking at two different things. Inside the cell, so we're looking at the membranes. This is the membrane. Inside the cell. Inside, outside. This is the membrane. That's that question. B, because it goes in to both directions and this direction, therefore it creates a sphere. Because it goes this way and this way and this way and this way, and to do that it has to be a sphere.
I figured it out from something he said. So it's rather than being the bilayer, it's the single span.
All right, let's come back together for a bit.
So that rather than being
What is the main function of a membrane now?
Philic, it's just philic phobic.
Out
And then why is the
versus
phobic on
in.
the inside?
That is it. You want to keep the inside in, the outside out. We can make it more permeable or less permeable by changing things. If I add things like cholesterol, I can change the permeability. Likewise, I could change the permeability by increasing or decreasing the length of the tails. How do each of those affect the permeability? So let's talk to each other. How do each of those affect the permeability?
Cholesterol lowers permeability by just like cholesterol gets stuck inside the membrane, so it lowers the ability of other things to pass through.
Oh, okay. So the cholesterol is of course inside, and then the hydrocarbon there, and that's a saturated.
So I think that's unsaturated because there's kinks, which means there's like
There's
double
kink.
bonds between the carbons.
Okay.
So I think all of these lower permeability.
But then you said that's unsaturated. So if it's unsaturated, it's
The
more
longer
permeable,
the
right?
tail,
Or not?
the harder the membrane is, the more rigid it is, the less permeable it is. If you have something really small, really short distance, you can jump that every once in a while. But if that distance is really long, it's going to be really hard to jump that distance.
Oh.
Okay? So the longer the tail, the less permeable it is. The more saturated it is, the more rigid, less permeable. If you are a plant that lives in the desert, say a cacti, how thick are your membranes?
Very.
Very thick, because you want to reduce permeability.
You want to reduce the amount of stuff that leaves
And enters,
and enters.
okay? Cholesterol does what? Reduces permeability. More cholesterol, more rigid. How big is your aorta? Big. It's the biggest blood vesicle in your body. It leaves the heart. That's your aorta. Okay? Do you want it to be flexible or not flexible?
Flexible.
You want it to be flexible. Because every once in a while, you need your blood pressure to do what? Go up. Okay? And if it is not flexible, can your blood pressure change? Not as easily. A healthy person should have pretty low blood pressure most of the time, but can have high elevated blood pressure and immediately returns back normal. That is fine. You get scared, fight or flight kicks in, you need to do something active, jump up and down, whatever it is, your blood pressure will go up. That's fine. You don't want it too rigid, because then what happens to your blood pressure?
It can't change.
It goes really high. Okay? So your garden hose, okay? A very flexible garden hose with a nice big diameter opening. How's the water flow? Beautiful. Great. Comes out wonderfully. High pressure, low pressure?
Low.
Low pressure. You make that garden hose rigid. Reduce the diameter a little bit, less flexible. What happens to the pressure?
High.
Goes up. What happens to the turbidity of it? Goes up. Turbidity means mixing. Do you want your blood to have mixing and high pressure? Do you want it to be low pressure, low mixing? Low pressure, low mixing. Okay? So, should you be concerned about cholesterol in your diet? Yes. Should you have cholesterol in your diet? Yes. Okay? You don't want too much or too little. Right? You want to be average. Right? The key for the next 14 weeks is to be average. That's our life goal. Okay?
So let's get degrees.
Yes. See
Next
good
question
degrees
for
or
you.
what?
Less permeable water. More Oh,
Less permeable.
less permeable to water. We only have to one answer though.
It's just one, right?
All right, 10 seconds.
Oh, wait.
But when 30-something of you already answered, I say 10 seconds. This is based on how many turned in. Most of you have turned it in, therefore I can say, talk to each other.
I don't know. I have no idea. Add more unsaturated tails to phospholipids. Lengthen the phospholipids.
More unsaturated is more permeable. So that's wrong. Lengthen the phospholipid.
Oh, less permeable. God.
Lengthen makes it.
I can do what to make it less permeable? Add cholesterol. I can also lengthen the tails. That's exactly what nature does. Okay? Want to make something less permeable to water? We're going to increase the length of the tail. We're going to make it more hydrophobic by putting wax on it. We're going to put cholesterol in it. That's how things work. Ultimately, we still want semi-permeability because we want things to be able to go in and out. Isotonic means what? Sane. Iso is sane. Hyper versus hypo versus inner versus out. So membrane is permeable to green solutes. What happens?
Becomes isotonic.
The green solutes move. Becomes isotonic. The membrane is not permeable to green solutes, but the water is still permeable. What happens?
The green solutes will pass through.
No, because the green solutes can't go through.
Okay, can't pass
The
through.
water will go through. The water level on this side will decrease and increase that side because you want to make it equal osmosis. We want to push things through.
So what was the last one, the correct one?
For which one?
For the last answer.
It was either tails or
Oh,
the
so-
cholesterol,
okay.
but it only let us answer one.
Yeah, it just allow us for one.
Yeah.
All right, talk to each other.
I picked B because the water inside the cell will try and leave to dilute the outside.
Oh, that makes
Which will
sense.
cause the cell to shrink.
That makes sense.
Can the green go through?
No.
No, but the water can. So what will happen? The water will leave, the cell will get smaller.
The cell will shrink.
All right, I have a question for you. Zombie apocalypse has happened. Okay, it's already over. Zombie apocalypse happened. You survived. Yay, congratulations.
Oh.
Your child is super sick and dehydrated and cannot ingest any water. You need to make an IV. Make me an IV.
Coconut
Design
water.
one.
Do we have coconut water? We have palm trees. He said make a what?
Make an IV.
Oh.
What can we add to a solution to hydrate our child?
Salt.
We don't want to give them too much.
Too much salt.
Because that would mean cells would
We need electrolytes, right?
What do you want in there? Do you want water? Yes.
Yep.
Want water because we want to put water in the child, right? What would happen if you put pure water in the IV bag?
It'll burst.
Burst. Water rushes in, and we just start blowing cells apart. Kid dies.
Yeah.
In fact, the kid would not die. It'd be exceptionally painful, and the artery would probably collapse, and the cells around it would collapse and fail. Anybody know why lethal injection in this country fails? Does anybody know why?
Fails?
It fails. It goes horribly wrong every time now when they try to do it.
They put water?
They don't have the right solutions done. Will any doctor or pharmacist or pharmaceutical company in this country make drugs for lethal injection any longer? No. You'll immediately lose your license. You will not do it. So lethal injection is not done by scientists. Who's lethal injection done by? The guards at the prison cells, at the prisons. They're the ones that make up the concoctions. They do it. Do they know how to make a correct IV?
No.
No. So when they do the injection, when you read, so go read about this. You'll know that the injections fail almost every time, and usually it takes multiple attempts now to execute someone. It makes it really painful and awful and horrible. But the reason why is they don't know how to make the solutions isotonic. And so what happens is I'm just going to put those medications in, and usually they use phenobarbital or concoction of, Dilaudid or opioids or something, which will do it. The problem is it's not isotonic. So as it enters the artery, what happens?
It bursts.
It bursts. The cells burst and die, and it fails, and it doesn't actually reach their system. So it never takes place. It never arrests the breathing or the heart rate. Well, you know better. So when you're saving your child with an IV, you're going to make it isotonic. So you're going to have what in it? Salt. A little bit of salt, a little bit of glucose, a little bit of sugars, a little bit of calcium. We're going to have about 0.8%. That's isotonic, is about 0.8%. And if we went to one way or the other, we can get cells to shrink or swell. So you want cells to swell slightly. So we're going to make it just a little bit more. Because we want to push the fluid into cells, but not do it to such an extreme extent that the cells are going to burst. Now you know how to save people, or do other things.
Do you think we have the right amounts?
Do you think you can calculate the right amounts? 100%. I just gave you the number, about 0.8%. So now you know how to calculate it. So, hell, you could all do this at home. If you needed to, right? You would get a sterile container. You would put salt water into it at 0.8% with a little bit of sugar. You would weigh it out, you make it perfect with versus volume. You would heat it up to such an extent that you would denature any enzymes in there and let it cool back down to body temperature. then you would inject it. You're working your way to be a doctor and scientist, that's how you do it. Right? Does anybody know how the first IVs went? Badly. But they knew that if they played around, they had just the right amount of salt, right amount of sugars, every once in a while a patient would survive. If you're going to die, or a chance of survival, is that ethical to move forward? You're going to die, but if I do this, you might die really quickly, or you could be okay. That's ethical. Today, we know how to fix this problem. Does anybody know how the first blood transfusions went? Either wonderful, or they died instantly. Today, we know blood typing. That is it. Okay? So, we want to always think about the ethics on our side. We have 150 years of science and medicine dictating it. We're at the beginning stages, guys. We're learning.
What's your
Okay? Sorry for sure.
I did too, but I'm not sure.
Well,
I know methane, but
we want the non-polar one, which would be methanol.
with a little bit of
Oh wait, methanol has
CO2.
What's the correct answer? CO2. Methane was really permeable.
Yeah,
Methanol,
nah.
not so permeable. The good thing. Anybody dumped methanol on themselves before?
Maybe.
Yes. What do you think nail polish and stuff is?
Methanol.
There we go. All right. What happened? You poured that on and it went through? Or alcohol, ethanol. Hey, just pour it on yourself. Ah. In fact, that does work. But not on all tissues. You have a waxy cuticle. Epidermal tissue does not allow ethanol to pass through. But certain tissues do let ethanol pass through. That you can figure out. You can Google that.
Say extremely. Wait, I got confused. Permeability, it means it protects.
permeability means it can pass through.
Oh, okay.
All right, talk to each other.
I think it's either B or D.
Yeah, that. Oh, okay.
It either can barely pass through or can't pass through at all. It's hydrophobic mostly.
Mostly
It only
hydrophobic.
has 2 oxygens. But it's very big.
Yeah, but it's very big.
But it's very big.
So I don't know, I put D. Let's just hope no.
Either B.
All right. Oh, I crashed it. What are we thinking?
Slightly permeable.
First of all, what molecule is that? Steroid. Which molecule of steroid?
Is it testosterone?
It's not testosterone, but a derivative of it. Progesterone.
Oh.
It's progesterone. Okay, so this is a steroid. Polar or non-polar? Very, very non-polar. So how many electronegative elements do I got? 2. How many carbons do I got? I ain't counting. Okay? So lots of carbons for very few electronegative elements. Therefore, this is going to be very non-polar. Therefore, this is going to move through very easily.
Very, very cool. Okay.
Very
How?
I guess we don't
It's
care how big it
big.
is.
However, it's just going to kind of bump up those membranes and be like, hey, likes,
That still has to
Like,
these are your steroids.
Yeah.
still has to
Yeah, more than
Yeah, derivative of it. Yeah, they're very. Those are your steroids. Got a funny story. Anybody know what blood poisoning is? Or anybody have blood poisoning sepsis? Be septic before?
I know somebody who is.
Okay. You don't want this. It's not pleasant. So if you get bit by a cat, cats have really terrible mouths. Wash it very quickly. Ex-girlfriend did not wash her hand after getting bit by a cat, and she gets home and I'm talking to her and there's a line running up her arm. And I was like, guess we gotta go to the doctor. So we go to the hospital. And making all the jokes aside, she's terrified of needles. Okay, absolutely terrified of needles. If you have blood poisoning, so it basically means the toxin is moving through your blood. And once it hits your heart, you're pretty much dead. So you got to get to it pretty quickly. So you need to build a solution of an antibiotic that is what? Fast acting and do what to your cells? Make it enter very quickly. Okay, so we need to make it enter very quickly. So we're going to shrink all those cells up. We're going to suck all the fluid in, and we're going to design it with essentially a high lipid. It's a fat shot. Okay, so she says to me, "Is it going to hurt?" And I'm like, "No, not going to hurt at all." Deep down, I know it's going to hurt like hell. This is going to be funny because this is going to hurt like hell. Anybody ever see those injections for septic?
No.
They're huge. You need a second needle to puncture open to inject Novocaine because the big needle is coming. Because it's so gigantic because you have to put literally butter into the person. You're putting so much oil with the antibiotic because you need it to spread immediately through the bloodstream and inject into every cell it comes in contact with. So is it going to be painful? Yes. Hell yes, it's going to be painful. She got mad at me for not telling her the truth. It's like, what am I going to do? Tell you you're going to run down the hallway and run away. Yeah, so don't pet cats. That's what the life story is. I got a kitty already. Yeah, I was showing people yesterday. I had a different kitty, and we had a kitty visit. This is a little kitty.
Oh.
So we had the big kitty a month ago, the medium kitty, and I got the little kitty in the house. So we're doing okay. Permeability rules. Steroids, non-polar molecules, gases move right through. Small, uncharged polar molecules can go through as well. Anything that's large and polar, rare, never going to make it through. Okay? So when in doubt, if it's rare in the environment, we're going to block its permeability. ATP is exceptionally rare in the environment, and it's very polar, and it's large.
Is there doing tomorrow?
No.
What kind of biomolecules are being shown here? Talk to church. What's everything being shown?
Well, I see the membrane. What are those green things?
Those are proteins.
yeah.
Those are
And
hydrocarbons.
those are like steroids. No, not steroids because they don't have OH.
They're hydrocarbons.
Hydrocarbons.
I mean, I haven't seen the shape before.
What's this?
Membrane.
Membrane, right? That's the hydrophobic. Then we have proteins and enzymes. These are Carbohydrate chains.
Carbohydrate.
Okay.
Yeah.
I have one last question. I don't have a slide for.
There's another question.
Oh.
Conformance and membranes. Rainbow trout.
Yep. All right, talk sure.
Not sure.
I'm not sure either. So they need to be able to adapt to temperature changes. Put more fluid, so an increase in unsaturated.
What do you think? No idea. No. What were you saying?
We can decrease the saturation or increase the unsaturated. That's it. All right. Re-exam is here. I will see you on Friday.
Oh, so it was either one of those two?
It was both decrease saturated and increase
Do this.
unsaturated.
Oh, okay.
Write up a reflection after.
At least I got one. But oh, and do you mind me taking pictures of